Okay, so I'm going to make this video just about reviewing for our lab practical. And remember, our lab practical is going to be on this Thursday, and it's going to include our microscope lab, cell structure and function, mitosis, and meiosis. Okay, so looking at the microscope lab, just to remind you guys about some of the things that we need to do, or that I'll test on. Definitely the parts of the microscope. I'll have microscopes out and I'll have different parts labeled, and you'll have to be able to identify the parts. So definitely go back to the page that we had to label all the different parts of the microscope. And then also, remember we had the table with the total magnification, this table, and we looked at the scanning power was 40x, low power was 100x, and high power was 400x. So I'll probably have a question on there where I ask you, I say if the ocular lens is 10x, then what would be the total magnification for the low power? And then you would have to look at the low power and you would see that it was 10x, so 10 times 10 would be 100, so 100x. So make sure that you know the total magnification or how to figure it up. And then we've already tested over some of this because we did the letter E and we had to make, you had to make your own cheek cell slides and stuff, so let's see. I believe that's probably it for that. And then for cell structure and function, we're going to have some questions about, remember you had to label the parts of the animal cell and also the plant cell. So what I'm going to do though is I'm going to have these models, and the model is going to show you or have the different parts. And so I'll have it labeled to where you have to identify one part, just one organelle that's in the model. So I'm going to go through really quickly. We have the nucleus, and then inside we have the nucleolus. We have the rough endoplasmic reticulum. And then the part that's the darker blue without the white dots on it, that's going to be the smooth endoplasmic reticulum. And then we have mitochondria, or these orange ones here. Here's the Golgi apparatus with the vesicles coming off of it. Those are the ones they normally will ask about. And then we also have our plant cell. Here, let me actually put it right here so it's better in front of the screen. We have the nucleus, the nucleolus. We have the rough endoplasmic reticulum, the lighter one with the white dots on it to represent ribosomes. And then the darker part's going to be the smooth endoplasmic reticulum. Golgi apparatus up here with the vesicles. We have the mitochondria. And then something else that we see that is not found in animal cells, but in the plant cells, we have the chloroplasts. So there's several of those. And then we have the central vacuole, and then we have the cell is surrounded by the cell wall. I did forget one thing on here that I wanted to mention for the animal cell is the centrioles. So plant cells don't have those, but the animal cells do, so I didn't point those out earlier. So I'll have a label on one of these organelles or structures, and then you have to be able to identify it. And then also remember that in this lab exercise, you had to be able to identify, or I guess not identify, but you had to know the functions of all the structures in the organelle. So make sure that you know that, because I'll have questions about that. And then go ahead and I think that would be it for that lab. Okay, then for the mitosis lab, okay, that one You definitely want to be able to know the parts of a chromosome. Remember, like the sister chromatids, the centromere. And you definitely want to know the phases of mitosis, obviously prophase, metaphase, anaphase, telophase. Make sure that you have them in the correct order. Then definitely make sure that for the animal cell and the plant cell, like here's the animal cell, definitely make sure that you can identify prophase, metaphase, anaphase, telophase. So, and also remember in the animal cell it's going to have the cleavage furrow, so I'll definitely have a station where you have to identify the cleavage furrow. And then plant cell, same thing. Make sure that you definitely can tell me or identify a cell that's in prophase, metaphase, anaphase, telophase. And so that right there is going to be 8 questions of the test. Is you'll have animal cell and plant cell where you have to be able to identify prophase, metaphase, anaphase, telophase. And then there'll be a couple questions, one where you have to identify cleavage furrow, and then for the plant cell, the cell plate. Right, then we did a little experiment in that lab. Make sure that for this lab that you will study the review questions for the mitosis lab. Definitely study the review questions. Okay, so then meiosis. I did have 4 slides set out as demo slides that were showing you the cells for meiosis. Sometimes I'll ask about those for bonus questions where I have it set up and I ask you what stage of meiosis it's in. So we definitely had metaphase 1, and I believe we definitely had at the end of meiosis where we had the 4 daughter cells. We had at the end of meiosis 1, beginning of meiosis 2, where it had the 2 daughter cells that were haploid. Seems like we had one more, but I can't think of what it was. And then definitely remember how we worked with the pop beads. So I'm definitely going to have some pop beads out. And, you know, if you have a pair of pop beads that are, you know, homologous chromosomes, a homologous pair, then I might have that out, like already set out with the pop beads. And then remember the magnets represented the centromeres. You may have to label that. And then remember that within one duplicated chromosome, this is a sister chromatid and that's a sister chromatid. But when you're talking about the homologous pair, then this would be a non-sister chromatid compared to another non-sister chromatid. So keep that in mind. It has them labeled here, non-sister chromatids. So make sure that you can identify those parts there if we're looking at a homologous pair of chromosomes, or if we're looking at a pair of chromosomes that are just like one duplicated chromosome. And then definitely just make sure you go through the questions that are in here. Definitely, you know, go through the questions about the human life cycle and make sure that you understand, you know, what is spermatogenesis, what's oogenesis. The testes are going to produce sperm. The ovaries will produce egg cells. Those will both be haploid, right? And then the zygote will be diploid. Make sure that you can compare meiosis and mitosis to each other. We had to fill out the table where meiosis has 2 rounds of division, mitosis has 1. The chromosome number in mitosis is, in the daughter cells, is 2N or diploid. The chromosome number in the daughter cells of meiosis, they're N or haploid. And then the number of daughter cells for mitosis is 2. The number of daughter cells for meiosis is 4. So definitely make sure you know that. And then definitely go through the review questions for the meiosis lab. Some of those questions I like to put on there. And that's really it for as far as studying that I think you should do. It's going to be 30 questions with usually 2 to 3 bonus questions. We all take the test at the same time, like we all start the test at the same time and finish at the same time. There's no talking during the test unless you raise your hand and you need some help from me, like you have a question. So if for some reason, like on the models, you can't find the label, or on the microscope you think something's maybe got bumped and it messed up, I can always come over and check it, and so you can raise your hand for that, but otherwise no talking. And you'll have your answer sheet that you'll fill out. You'll make sure that you keep it closed when you're not answering questions. And when you get finished answering a question, you'll close your answer sheet and you'll just be patient and wait until I say switch stations, because there'll be 1 minute per station. And so some students take the whole minute, but some students don't. And if it looks like everybody's standing and they have their paper closed, everybody's answered the question, I'll go ahead and I'll say switch stations. But I think that's it. Remember, we're going to start at 9 a.m. instead of at 8:30. So if you want to, we won't have class or anything. We'll just go ahead and start at 9 a.m. So remember, the lab door will be locked until that time. So then you can just wait in the classroom till it gets closer to 9 o'clock, or you can just wait in the hallway. And then at 9 o'clock, I'll come out and I'll pass out the answer sheet. And then I'll make sure that everybody, I'll go over the rules again just to make sure everybody knows. So please email me if you have any questions. All right, thanks.